Click for IB Biology. Here we're dealing with polynucleotide chains. There's a blank PDF of this presentation down in the description that you can use to annotate for your own notes. We have 10 quick questions for you, and we've provided the answers so that you can test your understanding of this content. And then at the end, in the channel links, we'll show you the link to the next video and indeed to the playlist and to the channel. So as the name suggests, polynucleotide means that we're going to be joining many nucleotides together, and we're going to look at the mechanism for how this actually occurs. So we've got the first polynucleotide is here and the second polynucleotide is here. So we're adding number 2 to number 1. The link between the first and second nucleotide occurs through the phosphate group on the 5' and the 3' sugar, an OH group on that particular sugar. The bond that we're going to form is called a phosphodiester bond. Like all of the bonds that we've been discussing so far, this is a covalent bond. And so on this side here, we can see that we have, this is number 2 and this is nucleotide number 1. And so we've got the 5' here is now connected to the 3' on the first sugar. So we start to build up the sequence of 3', 5', 3', 5'. Notice that the bases are pointing towards the center here in this way. This particular reaction is often characterized by the phrase 5' to 3', referring to the fact that the second sugar is added to the first sugar's 3'. And this in DNA is the function of the enzyme known as DNA Polymerase 3. This is, by the way, is a condensation reaction. In the polymerization of the chain to form an RNA chain, the enzyme involved is RNA polymerase. And so now, having joined the two nucleotides together, we have what is actually called a dinucleotide, small point really, and any subsequent additions will occur at this position here. So if we have a third nucleotide coming in, it will be attached 5' to 3' at this position here. So number 3 nucleotide would add at this point, not at this point here. If we look at a chain which I've drawn to be slightly longer, we've got 3 nucleotides in our chain so far, and I'm about to add a fourth, so again it's 5' to 3', so 5 to 3. That's the way this has to be. That's the way in which the enzymes add. They never do so in reverse. And so notice that what we've developed here is a sequence of phosphate, sugar, phosphate, sugar, phosphate, sugar, down into what is now called the sugar-phosphate backbone of the polynucleotide chain. The description I've just given you applies to both DNA and to RNA. The bases are sticking it into the center here like this. Links to the next video on DNA is appearing here, along with links to the channel and indeed to the full playlist.